Hello again. It gives me great pleasure to introduce our keynote speaker, Dr. Becky Schweighardt, who is Chief Scientific Officer and Chief Operating Officer at Grace Science, based in Menlo Park, California. Dr. Schweighardt is a trained immunologist and virologist who specializes in disease pathology. Dr. Schweighardt received her PhD in the laboratory of Professor Walter Atwood at Brown University, working in the field of HIV, before completing her postdoctoral fellowship at the Gladstone Institute of Virology and Immunology. She is an experienced translational scientist, dedicated her life towards all stages of development, drug development, from discovery to post-marketing and across multiple drug modalities, from gene therapies, protein therapeutics, oligonucleotides, and small molecules. Dr. Schweighardt has over 25 years' experience in academia and industry, including immediately prior to joining Grace Science in 2020, 10 years at BioMarin, helping develop rare disease for patients there. Please join me in welcoming Dr. Schweighardt to the podium for what will be a fascinating and I think challenging discussion on the development of gene therapies for ultra-rare disease. Thank you, Dr. Schweighardt.
Good morning, everybody. I'm so thrilled to be here, and thank you to the organizers for inviting me to give this speech and to really tell you all about Grace Science. So we can move forward to the, I guess I can move. I'm going to start with talking about the ultra-rare disease that we work on at Grace Science. So the disease is called N-glycanase 1, or NGLY1 for short, so you'll hear me saying that. It's a severely debilitating, ultra-rare disease with a limited life expectancy and no approved therapy. N-glycanase 1 is an enzyme, so it really is an essential enzyme all over the body, but really impacts the neurodevelopmental system greatly. So the core phenotypes you can see are there: global developmental delay and intellectual disability. There's also hyperkinetic movements, transient elevations in transaminases, and hypoalachroma, which is it's an inability to make tears, which causes a lot of problems in the eye, and severe neuropathies. And you can see a long list of other phenotypes: of failure to thrive, cerebral atrophy and microcephaly, epilepsy and seizures, again, the eye problems because of the alachroma, feeding difficulties, and so on. So the average age of diagnosis is about 6.5 years of age, which might seem kind of old for children that have such severe disability. And many of the parents will tell you that they were able to tell in utero that there was a problem. But because it's such an ultra-rare disease, the patients often go misdiagnosed. Sometimes they are diagnosed with other diseases. So it takes quite a while. But thankfully now, with different methods of whole genome sequencing, the patients are getting diagnosed earlier and earlier. What we found at Grace Science Foundation is that the mean age of death is about 15 years of age. This was a newly identified disease, so we're really developing and learning this information all the time. So it's difficult to say in words about what this disorder does to these patients. I have a video that I'll show after so you get a better sense. But basically, NGLY1 requires constant daily care for all aspects of living: feeding, bathing, toileting. Most of the patients cannot walk unassisted, and nearly all patients are nonverbal. There's physical occupational therapies, speech therapies that patients will do all the time that help them. There's also many patients require G-tube surgeries to maintain adequate nutrition. And the NGLY1 patients often present with different medical issues that require frequent hospitalizations, including their seizures and chronic infections. They also have many surgeries, spinal fusions, tracheotomies, and for the G-tube, and of course, as I talked about, the shortened lifespan. So there are about 100 patients that we know of worldwide that are living. There's about 15 patients that we have tracked over the years who have passed. This seems like a very small number, and it is, but that number grows. Every month we are talking to new patients. They reach out to the foundation. A lot of times the parents will find out, they know that there's something that has been wrong with their children for years. They will go and they will get the sequencing done and find out their child has NGLY1 deficiency, and then they don't know anything about that. So they go on a long journey, and thankfully the Grace Science Foundation can help them on that journey. So if you've noticed, I keep saying Grace Science Foundation and Grace Science the company. Those are two different entities. So the Grace Science Foundation was started by Matt Wilsey and his wife, Kristen, for their daughter, Grace, is the namesake of Grace Science, has the NGLY1 deficiency disease. So they started the foundation back in 2014 when their daughter was diagnosed. And they started having different conferences and funding different researchers, and they've learned so much about the disease. They started a natural history study at Stanford that went on for 3 years, and they have just started a new natural history study that's focusing on the movement disorder at Baylor College of Medicine in Texas. So the Grace Science LLC, where I work, was co-founded by Matt and Nobel laureate Dr. Carolyn Bertozzi. And really, we focus on an AAV9 gene therapy for NGLY1 deficiency. And we also have research programs, the small molecule research programs that are based on more common indications on the NGLY1 biology and pathway, so different targets in the pathway that we've really learned so much about. That's true serendipity from this terrible disease. So I'm going to show you a video because that's probably an easier way to see the patients than for me to talk about them, so you can really understand this. That really talks about the Grace Science story. The foundation, early on in the foundation days, we worked with Takeda and Taysara in Japan at the Riken Institute, and Tadashi Suzuki, who developed the animal model of disease, which is an NGLY1 rat model. So that's what this video is about. So I'll just step back for a minute so you can watch the video.
Grace is a sweet, lovable little girl. She's 10 years old. She is fascinated by music and books. She loves to eat. She's very, very social. She has an ultra-rare genetic disease called NGLY1 deficiency. There's less than 100 that have been identified in the world so far. So this is one of the rarest diseases known to science and medicine. NGLY1 is a global developmental disease, so it basically affects every system in the body, primarily the brain. So while she's 10 years old, she really acts almost like a 2-year-old. And she can barely walk. I think communication is probably the hardest facet of the disease. Knowing the enemy is so valuable, not knowing who we're actually fighting. It's confusing. My background is a tech entrepreneur. When Grace was diagnosed, I shifted gears fully to dedicate myself to understanding this disease, treating it, and eventually curing it.
希少疾患の患者さんとつながりを持つことは、研究者のモチベーションに大きく貢献します。
今までは本当に病気につながるということを考えずに研究してたのが、まさか患者さんのための研究をすることになるのかということは、特別な感情といいますか、全く思ってもいなかったことなので。
One of the best, most successful therapies for her so far has been working with horses, where she'll ride a horse with a sidewalker. And when she first started, she barely could put her legs on either side of the horse. She was really a little peanut. Now they have her, with assistance, riding backwards, riding sideways, and it strengthens her core tremendously, which is so important for everything. My hope for NGLI1 deficiency patients is that they will lead an independent life, and they'll be able to do simple things like bathe themselves, dress themselves, feed themselves, maybe hold a job, live on their own. Those are our long-term goals for independence.
我々が今経験している中で一番難しいことっていうのは、どうやってやっぱり患者さんに早く薬を届けるかっていうことですね。
希少疾患の新しい治療法に取り組んでいる皆さん、たくさんの患者さんが首を長くして研究の進展をお待ちです。一生懸命頑張りましょう。
Just takes one little spark to unlock a disease, and you can do that. The more brilliant minds working on this, the faster we'll get to a solution.
So now you understand the disease a bit more, and I hope what that brings to you is the closeness of the work that you do every day and how it can impact the lives and families. It's very close to what we do, and I feel very grateful that I get to work with the family. I've worked with rare disease for many years. I worked at BioMarin before. I worked at Grace Science, but it really is an honor to be able to work that closely with the families. So I will start talking about NGLI1 itself and the pivotal role it plays in degradation of glycoproteins. So NGLI1 itself is a cytosolic enzyme that cleaves N-glycans from misfolded proteins. So it facilitates their degradation by the proteasome. So without NGLI1, it really disturbs the proteostasis and results in the accumulation of cytoplasmic ubiquitinated proteins. The alternate cleavage of N-glycans leads to the accumulation of GNA, which is a biomarker of NGLI1 deficiency, and that can be detected in the CSF, plasma, and urine of the NGLI1 deficient patients. And if you look here at the top part of this graph, that is NGLI1 being able to cleave the N-glycans from the proteins, and then the proteins are able to be degraded by the proteasome. But then along the bottom there, if NGLI1 isn't there, you get an alternate cleavage, and you get that little glick neck that's stuck to the amino acids that we can detect by mass spectrometry in the CSF and plasma of NGLI1 deficient patients. And this is showing the increased levels of GNA. So over on the that's your left. You can see the CSF in the plasma of NGLY1 deficient patients, and you can see that it's increased in the NGLY1 deficient patient, and the control samples, it's undetectable. And then also I mentioned the NGLY1 deficient rats that Tadashi Suzuki's lab developed, this over on the right, and you can see in the CSF and in the plasma of the rats, we're able to see increased GNA levels as well. And what's interesting is in the heterozygous animals, which are in the center of the graph, the GNA levels are slightly increased compared to the control animals, the wild type. So the NGLY deficient rodents have severe neurological symptoms. So I'll show you two videos. One is on the right of the NGLY1 deficient animals where they have a gait abnormality, a hind limb drag, and a tremor. Very similar to the movement disorder that we see in the patients. And then you can see in contrast to the wild type animals that have a fluid movement, rapid acceleration, they're exploring, so very different symptoms in the NGLY1 animals. And then over on the right, you can see in the black line, this is latency to fall from a rotor rod, so the animals are put on a spinning rod. And they are able, the wild type animals are able to stay on that rod quite a bit longer than the NGLY1 knockout animals that are in the orange line on the bottom. So this chart is showing phenotypes that are seen in the humans that on the left and in the NGLY1 rats, and how we can look at differences in the rat model that mimic the humans. So both, have a failure to thrive in the humans, and that reduced survival and fitness can be measured in the rats. The humans show gait abnormalities, hypotonia, and peripheral neuropathies. These can be measured in the animal model as well as when we do the necropsies. You can see axon degradation in the spinal cord. You can see degradation in the sciatic nerves and in the dorsal root ganglion. And in humans, you see this intellectual disability, delayed cognitive development, the lack of language development. And in the rats, we're able to measure impaired spatial learning, and we can see that neurodegeneration upon necropsy. And as I mentioned, we also are lucky enough to have a biomarker, which is a pharmacodynamic biomarker of the disease, increased GNA that can be measured in the CSF and the plasma. So NGLY1 deficiency itself causes neurodegenerative pathology, and that can be seen here. In the left panel are wild type brains, and in the right panel are the NGLY1 deficiency, and you can see necrotic lesions in the NGLY1 animals, where you can see that there's neuronal death and astrogliosis, microgliosis. The brain regions that are affected are the thalamus, the hippocampus, the medulla, cerebellum, and pons, so the deep structures in the brain. And in the sciatic nerves, you can detect axonal degeneration. And in the dorsal root ganglia, or DRG, you can see necrosis of the cell bodies, neurodegeneration, degeneration of the nerve fibers, and infiltration of the mononuclear cells. And also, we can detect in patient autopsy samples very similar findings in the patients, including eosinophilic cytoplasmic inclusions, so those inclusion bodies, which are likely the conglomeration of the proteins in the thalamus, spinal cord, and the DRG, as well as widespread Purkinje loss in the brain. So as I mentioned, at Grace Science, LLC, we are developing GS100. It's an AAV9 NGLY1 gene therapy for NGLY1 deficiency. It's a recombinant single-stranded AAV9 vector that encodes a codon-optimized full-length version of NGLY1, the human NGLY1, and it's under expression of the CAG promoter. And this is delivered in the rat model, and this will in the clinic as well, through intracerebral ventricular or ICV administration. This is basically goes right into the ventricle and the brain, so it's delivered into the CSF. And NGLY1 deficiency, as I've mentioned, is primarily a disease of the central nervous system, with the brain being greatly impacted by neurodegeneration. So it makes sense to deliver to the brain. In the non-clinical studies in the rats, ICV administration resulted in improved motor function and reduction of the GNA biomarker when it was delivered to the brain, whereas IV administration didn't result in the same improvements. ICV administration delivers the drug to the proximity of the areas of the brain, those deep structures that are most impacted by NGLY1 deficiency. And ICV administration is commonly used for delivery of pediatric therapies for enzyme replacement therapies, antibiotics, and chemotherapeutics, as well as some gene therapies. So many of the neurosurgeons are used to the procedure. So we measured DNA and the vector DNA and the NGLY1 mRNA in the rat model of disease. So you can see here on the top is the vector DNA, and on the bottom is the human NGLY1 mRNA in the brain on the left and the spinal cord on the right. And you can see a dose-dependent increase in vector DNA that was detected in the NGLY1 animals, and also a corresponding increase, dose-dependent increase in the NGLY1 mRNA that was detected. And then we also looked for the actual protein expression by immunohistochemistry, and we were able to detect widespread human NGLY1 protein expression across the rat brain. And the vehicle control, it's kind of hard to see, but there actually is a picture there. But you can see that there's lots of dark staining of the NGLY1 protein in the treated animal. And then we also are able, this is looking at the GNA detection. And here we have CSF on the left of the brain and the spinal cord on the right. And here you can see the vehicle-treated animals are in orange. You can see that the GNA levels are high in the vehicle-treated animals. And then we have a dose-dependent decrease in the GNA across the tissues and in the CSF. And over there on the right, for each of the graphs is wild type. So we're not getting down to the wild type levels, but we're getting like a 30 to 40% decrease in the GNA in the NGLY1 animals. And interestingly, we have a very strong correlation between the GNA levels that are detected in the brain and in the spinal cord with the CSF. So that will be very useful in the clinic, that we can measure the CSF levels as a surrogate for brain tissue GNA levels. And you can see here, these are correlation plots with the brain tissue GNA versus CSF GNA on the left, and on the right is the spinal cord versus CSF GNA. And you can see strong correlations and drug dependent, where at the higher doses we have lower GNA. And here we're looking at the motor function. So this is showing a picture of the rotor rod test, where the animals are placed on the spinning rod to see how long they can actually balance on the rod before they fall. And you can see in orange is the NGLY1 knockout animals, so they're not able to stay on the rod very long, versus the wild type animals, which are on the right in black, where they're able to stay on much longer. So what we can see is a really nice correlation, a dose-dependent correlation, where the animals are able to stay on the rod longer, so they have more strength and coordination once they're treated with the NGLY1 deficiency, and when they're treated with the GS100, and it gets to almost the wild type levels at the higher doses. And what we also see is that there's a really strong correlation between the GNA reduction in the CSF and the brain tissue with motor improvement. So here we're looking on the left is rotor rod latency versus CSF GNA, and on the right is rotor rod latency versus the brain tissue GNA. And you can see strong correlations. And the different colors there are the treatment. Again, the orange is the vehicle. The black is the wild type, and then you can see the dose-dependent response there. So this is really important for the clinic when we have a pharmacodynamic biomarker like this, where it's correlating, and you might say predicting, improvement in the animals. Hopefully we'll be able to see that improvement in the clinic as well. And it's possible to, the Sarepta gene therapy that got approval a couple of weeks ago, that was based on dystrophin, which is a biomarker protein expression from the gene therapy, that they were able to get approval on that accelerated pathway. So the conclusions from what I've showed you about GS100, the treatment resulted in widespread dose-dependent vector distribution, mRNA expression, and NGLY1 protein expression across the brain and spinal cord that correlated with the biomarker reduction and motor function improvement. There was a strong correlation between the CSF GNA and functional improvement in the NGLY1 deficient rats, which suggests that GNA will be useful as a predictive pharmacodynamic biomarker in the clinical trial. Also, what we found was GS100 was tolerated well, and we didn't have any drug-related systemic toxicities in the NGLY1 deficient animals. But we did detect a dose-dependent mild to moderate neuronal cell loss in the GS100 treated rats that was close to the site of injection. We believe that this was due to the spatial constraints of dosing in the 28-day-old rats. These are very tiny rats, and the needle is quite large in comparison to the brain size. Thankfully, the findings were not associated with any worsened functional outcomes in the animals, and we don't expect that this will occur in the clinic when the drug is administered by slow infusion through a catheter placed into the much larger ventricular space in the humans. However, I can say that we submitted the IND based on this data, and the FDA had concern over that neuronal death that was detected in the animals. And they requested that we do a larger animal study before going into the human trial. So we triggered an NHP study almost immediately. $2.5 million later, the animals were fine throughout the study. They came down last week. We had no drug-related deaths, no abnormal clinical chemistry of macroscopic findings, thankfully. But the histopath is ongoing, so we will resubmit in July, at the end of July. So that's our story of gray science. I thought I'd try to bring it back to the bigger picture and how this impacts rare disease and ultra-rare disease drug development in general, because we have found a lot of pain points along the way, and I'm sure many of you have as well. And I'm sure that many of them are a lot of the same pain points. So I wanted to talk a little bit about the challenges, but then I'll also talk a little bit about the opportunities where this can actually help us move things forward. So of course, challenges with ultra-rare disease, in rare disease in general, when there are so few patients, it's really finding enough patients to recruit. So, you can be searching all over the place, but really being able to find patients who fit the inclusion or exclusion criteria for a trial can be very challenging, particularly when, you're being asked to have specific inclusions or exclusion criteria based on safety. You want to have the safest trial as possible, but it limits the number of patients from a very small population to begin with. And of course, you want to have an accurate diagnosis. Many patients with rare disease, as I showed you, are often misdiagnosed or they're undiagnosed for multiple years. So thankfully now that we have more genetic testing that's ongoing, patients are being diagnosed accurately, but still many of them have sequences of unknown variants. We don't know if they're actually a pathogenic variant, so it really has to be correlated with the symptoms. Thankfully, we have a biomarker, so we can test the biomarker in the patients, but it is a challenge with rare disease. And patient location. Often the patients are globally dispersed, so you have to have multinational trials that can be challenging for the patients for travel. Many of them are in remote places. And then, of course, designing and evaluating the clinical trials themselves. It can be very challenging to characterize a disease when there are less than 100 patients worldwide that have that disease. And of course, there's limited natural history study. We're dealing with this now. The Grace Science Foundation was the entity that really performed the greatest amount of put together the greatest amount of natural history study. But it's limited. We have less than 4 years of natural history study for this disease. So do we not move forward with a therapy that we know is treating the disease because we need to understand more about the underlying disease? These are some of the challenges. Also for statistical analysis, of course, because there's heterogeneous manifestations of disease, and phenotypic presentation can be very diverse across the patient, so that's really challenging when there are so few patients with the disease. And then to have a proper comparator group in the trial. Placebo control is often impractical, if not impossible, with rare disease and ultra-rare disease. And again, the natural history data for having any kind of comparative data is often sparse, so it makes it very challenging for the trial. And patient burden, designing rigorous trials that have enough flexibility to limit patient burden, but have enough stringency so that you can actually analyze the trial robustly. And then the challenge of multiple regulatory regions. And I'm sure many of you have felt this pain of trying to harmonize trials across different countries, which can be very challenging when the different regulators are asking for different things. And you have one protocol with a very few number of patients that you're trying to harmonize across those patients, and also different standard of care across different regions. For regulatory approval and payer reimbursement, collecting and communicating evidence that's convincing and compelling to regulators and payers is very challenging. And that's due to the limited amount of data and the inability to run traditional randomized placebo-controlled trials. So for manufacturing, I thought I'd highlight this one since I'm not a manufacturing person from the get-go, but wow, have we really hit a lot of challenges in that area. So I wanted to talk a bit about that. First, of course, the high cost. It's an astronomical cost of goods, the raw materials, the extensive amount of testing, the number of subject matter experts that you need, and the service costs. And there's limited commercial opportunity for the rare diseases, of course, because there's a small number of patients. For process development and the GMP manufacturing itself, the available processes are complex and they're not standardized across the field. And the PD studies are expensive and time-consuming. So there's a tendency to really stay with what may be a suboptimal status quo because that innovation comes with a lot of risk and cost. And it's challenging really to determine what's a critical process parameter when you have a limited number of batches. You know, you may have just 2 or 3 batches for the entire patient population. How can you determine what's critical to measure among the limited number of batches? Analytical characterization, there are challenges with assay development and validation timelines, accuracy, reproducibility. I'm sure I'm preaching to the choir here, but there is no one-size-fits-all standardized testing approach. The phase-appropriate challenges for ultra-rare disease when the first study may be the pivotal study that's used for registration. So you don't have the time to test these things out. And then you need large drug volumes for the testing. And this reduces the amount of drug product available for the patients and drives up the cost. I can say from our own experience, almost 50% of our first batch went to all of this testing. And we weren't even sure why we needed to do it all. And I'm not sure we needed to. But we have patients that are in line to take this drug that are waiting, and they won't be able to because we have buckets of stability testing ongoing and sterility testing ongoing. For comparability, it's especially challenging with, and I know you spoke about comparability yesterday at the symposium, it's particularly challenging with the limited number of batches that are needed for these rare disease trials. So those were all the bad things, but there are good things from opportunities that come from rare disease drug development. So it provides really the unique challenges that are there, and it's why I love rare disease development. I mean, it's not only the purpose of being able to work with the patients, but it's the creative problem solving that comes from it. It really drives innovation. You have to really blaze your own path. There's no map to follow. Thankfully, rare disease drug development has led to really streamlined processes for specialized regulatory pathways like fast track designation, priority review, accelerated approvals, really exciting changes. And of course, the US Orphan Drug Act really defined rare disease and provided companies with financial incentives to pursue drug development, and that has led to lots of companies working on different drugs for rare disease. And rare disease has already spurred the formation of specialized funding programs and different infrastructure, including the Bespoke Gene Therapy Consortium, the NIH Rare Disease Program, and multiple rare disease grants and funding opportunities that are out there. Patient advocacy in general, the media coverage of the disease severity, these drive a very passionate call to action. And it's a sense of urgency that's created that has that strong push for innovation. When people are feeling emotion around something, they'll often help you to solve the problem and possibly even pay for some of that problem solving. So one of the biggest opportunities, I think, also is the opportunity to share information freely when there's not a direct conflict of interest. You know, if we're not all working on the same indication, we can share information and hopefully learn from each other. So that's how I will close my talk today. And thank you again for listening and to the organizers for bringing me here. I really feel that there are opportunities for expediting and reducing costs of ultra-rare disease, and a lot of that will come from information sharing. So being at conferences just like this one, where we each hold a puzzle piece that if we talk to each other and share that information openly, we can solve problems together that we can't alone. For regulatory agencies and companies should really be publishing meta-analysis for across tox studies. You know, for this, it's really the objective to help the field identify and differentiate between, expedited, expected AAV class effects, what's a species-specific effect, what's a procedural effect, so that companies aren't having to do expensive non-clinical studies that aren't necessary or helping the clinic. And also for AAV manufacturing collaboration and information sharing, more open collaboration and information sharing between CDMOs and sponsors and regulatory agencies to publish meta-analysis and white papers that share blinded AAV9 batch information across programs. The objective here being to help focus the field on the critical quality attributes that require continued testing, to define what's a standard residual impurity range, and what's an expected AAV stability range, so that we don't have to spend half of our drug product on testing, and we can spend them on our patients. So with that, I'll close and thank you all for coming. It's been it's wonderful to be here at my first CAS meeting. So thank you.
Thank you very much for the presentation there, Becky. It was very, very interesting, and certainly the first part was, I thought, quite emotional. Going to the development of GS100, oh, there's working now for sure. I'm curious, why did you select AAV and AAV9 in particular right at the start of the story, and when did that journey start? Yes, it's on. Yes.
Okay. So we chose AAV9 because it had been used in other CSF administrations for other neurological diseases. So that was, we didn't really, we didn't want to be the first to use, something that was very innovative. I'm just going to slide over here so I can see Kathy a little bit. So yeah, that was really why we started with the AAV9, and it was many years ago, as you can imagine. We started our proof of concept studies with AAV9 back in 2015. So it took a long go of it because they had to get the vector right, but that was why we chose AAV9 in general. And there are approved therapeutics for AAV9 as well. So there's certain trails that we have to blaze, and then certain ones that we have to make sure that we're doing something that others have done before us, and that was one of them.
Mm-hmm. And did you explore any other modalities of enzyme replacement, recombinant protein therapeutics, for example?
Yeah, so that was before my time at Gray Science, but of course I worked on many enzyme replacement therapies at BioMarin. Because it's a cytoplasmic enzyme, it's very challenging for enzyme replacement, so the gene therapy was the better modality.
Thank you. I think we'll take a question from the audience.
Hi, Taro Fujimori from Ultragenics. Thanks for the excellent talk. Kind of following on to Rob's question, the AAV9 and looking at the locomotor skills kind of maybe addresses one of the many symptoms that the disease has, and you mentioned that the enzyme is very widely distributed. So curious if there's any thoughts about if you are going with AAV9 to also combine it with some other serotypes to hit other locations.
So we actually, when we do I didn't show this data, but when we do the biodistribution data and the expression, we're actually getting quite a bit of expression in the periphery, in the heart, in some in the liver, different muscle tissue. So we are getting pretty good expression in different areas, even with the AAV9.
Okay, thanks.
If I could ask one more question, actually, I've got several here of my own, so I'll be a bit selfish. Ask one though, and then go back to the audience and online. You mentioned, in the video, speed to the clinic is really critical for these patients. There's patients waiting, families waiting for this treatment. What do you think are the top one or two aspects that maybe hinder that speed to market, that you think, that there's potential to do something there but still maintain the scientific integrity of the study?
Yeah, that's a great question. And I have to be very careful about the way I answer that.
Okay.
In hindsight is 2020. So it's really easy to say what we didn't have to do versus what we have to do. The $2.5 million NHP study was a bit hard to swallow for us. So there's not too many there's not too many companies who could have raised the kind of money that we had to raise in an ultra rare disease to do that for something that, we're not seeing any issue in the animals. So, that's a place where we need to get better at sharing information so that unnecessary non-clinical studies are not done. So that's probably the biggest one that comes to mind right now. Also, regulatory, being able to really expedite the pathway. We speak with Peter Marks all the time, and he has been so amazing at helping us. So there are opportunities there for the FDA to really work with the sponsors, and thankfully we have had such good luck in speaking with them, and they help us all the time. But I often worry if sponsors aren't reaching out, then they won't be getting that information, and we are the squeaky wheel, so we reach out often. So I would urge people to do that.
And then a follow on from that, you mentioned a lack of sharing of, for example, preclinical models and some of the challenges there in maybe a generic or de-identified way. Were you aware, for example, of mouse studies also using the intracerebral ventricular device that also caused neuronal death, for example, that didn't go on to in larger animals that you could have leveraged to reduce the reliance on NHP?
Yeah, we did try to do that, and thankfully, what has been so amazing since I've started working at Grace Science is how so many ultra rare disease companies and rare disease companies will share information with us. And I think a lot of that has to do with our CEO being a parent. So when Matt reaches out to somebody, they really want to help. So we have a lot of information that's not even published from other companies where they have done ICV administration in rodents and seen similar things. The concern, to be fair, the FDA was concerned that it was dose dependent for us. So it's likely because it's the site of an injury where we were getting leakage into that area, but that we can't prove that until we see a larger animal. They did not ask us for an NHP study. They asked us for a larger animal study, so we just had the NHPs ready to go because we had them contingently planned. So I would say that's a good thing to do, is to have mitigation and risk contingency planning, because that's the only way that we are where we are now, is because we had suspected that would happen.
Online questions, Kathy?
I think there's a...
Sorry, I'll take a question in the room first of all, sorry. Didn't see you there.
Hi, thank you. Shannon Holmes from Ultragenics. I had a question about the device delivery. So you mentioned you'll have a catheter in the ICV space to deliver the drug. In your interactions with FDA or other regulators, have they asked you to do any particular types of device drug compatibility studies, and can you share what those are?
Yes, they have asked us to do the device compatibility studies, and we have done it in all the animal studies to make sure that the amount of dose that we were putting in was exactly what we thought we were going to deliver. And they have specifically asked us, the FDA has specifically asked us multiple times to do the device compatibility study for the clinic, which we have already done. But honestly, we had challenges with that, and we had to reach out to many people. What does that look like? How do we plan it? That's one of the things that's sitting in my email right now, is to review that device compatibility study. So I'm hoping that we did it correctly, but there's not a lot of guidance out there on that. So that would be an area. You're probably talking about that, I think, tomorrow, or maybe it's today. So I'm going to attend that session. But yeah, that is something that's very challenging.
And then a follow-on question for that. Did they ask you, or have you looked at the endotoxin contribution of your devices as well as your drug? And have you had challenges getting the dose high enough, yet ensuring your endotoxin is not over the limit for an intrathecal type administration in smaller patients?
Yeah, that's a great question and a really challenging one. So we're under the endotoxin limit, but that was challenging. So that wasn't something I talked about here, but we are at quite a high concentration. So we're at a 1.5 e to the 14 VG per mil concentration. So that issue was something that the FDA called out specifically to us. And thankfully, we're underneath it. But that is something that needs to be thought about when you're delivering directly to the CSF. It's a lower limit of endotoxin. And when you start to get up for these CSF administrations, if you need a high enough dose, that concentration issue becomes a problem. Maybe I'll diverge a little bit from your question, because what I have found from talking to people about the concentration that we went at, so many people told us that we would not be able to concentrate that high, and we have, and it's stable, and there's no aggregation, so it can be done.
Thank you so much.
So I thoroughly enjoyed your talk, Becky, and it illustrated so clearly the challenges with ultra-rare diseases and challenges that go across many different CMC aspects. The one of them you mentioned was about you're producing a product in small amounts, and we've also worked on products where we use more of a drug product batch to do testing than we put into patients. And this is really one of the areas where we need to continue to drive, even in analytical methods, to use smaller amounts, particularly like sterility testing, to get down to really small amounts of the product required for testing. So, but overall, when you were explaining all of this, it just calls for leveraging platform knowledge, right? And we've seen the recent US legislation calling for the use of platforms, and a lot of discussion about that. This is a really, really nice example. I wondered, is Gray Sciences involved in the Bespoke Gene Therapy Consortium? Are you able to leverage the consortium?
We are not involved in the Bespoke Gene Therapy, and that was because we were far enough along once it started that it was kind of a moot point for us. But I think it's amazing that has started. I applaud that, and I hope everything. We are so open to sharing information. And I think that is the way to go. So if they can leverage the information that they're learning from the Bespoke and share it with other rare disease companies, I think it's fantastic. But no, we haven't leveraged it.
One thing you mentioned that was perhaps a challenge was divergence of guidance across regions. So have you had interactions with health authorities outside of the US, and if so, what were the major differences between USFDA and those countries?
We have talked to other regions. We're not as far along with the other regions. We have spoken to the EU and to the MHRA. We'll be speaking with Health Canada soon. Interestingly, it was mostly about the device. So that is interesting. They were pushing us, wanting us to go ICM. So in intracerebral, to the base of the brain, into the, I can't think of that word while everyone's looking at me, but, and so it was very strange that they wanted us to do that, because our neurosurgeons were telling us that it was a greater risk because of just the physiology of the patients. But that was something that we continuously heard. They were really concerned about ICV as an administration procedure. And of course, I was at BioMarin before I came to Grace Science, and Brineura, which is for CLN2, Batten's disease, we administered repeatedly an enzyme replacement therapy through ICV. So I think it's just a matter of people getting used to that administration procedure. But yeah, other than that, there wasn't a lot of big differences, except maybe clinically for endpoints. There was some different endpoints that each of the regions wanted. So I can imagine that will be challenging for us moving forward. Everybody has their specific endpoint, especially for neurocognitive endpoints of what we should measure. So I'm not exactly sure how we will iron that out moving forward. We were hoping to be able to move forward on the accelerated pathway with our biomarker, since it's a pharmacodynamic biomarker, but I can say unanimously that came through all the regions, is that we have to be able to prove it, because there's no, we have to be able to prove that there's clinical relevance to that GNA biomarker, which I understand.
Mm-hmm. And on that note, you got both a device and a biomarker. So have you also had to have discussions with the Center for Devices and Radiological Health as well at FDA, or has it primarily been through CBER?
No, it's primarily been through CBER. So, but we would have welcomed that, really, because it was challenging. Yeah, and I have to remind myself all the time to ask, because when something is challenging, honestly, the FDA has been so helpful with us that when we do ask, we do get answers. So we probably, in retrospect, should have asked that question in more detail about the devices. But yeah, we haven't. We've just been going through CBER.
No, thank you for that.
Yeah, there was a fairly specific question that came in online, I think, about the formulation that you used when you went to the higher concentration of the product, and managed to do that without seeing aggregates. And so the question is whether you could speak a little bit more about the formulation that you used.
Am I going to get this right? That's the question. Where's my CMC expert when I need her? We have a phosphate buffer. I think we have paloxamer in there.
Those sound right.
Okay. Thank you. That's all I can remember.
That's great. I think we're actually at time now. It's 10 o'clock. So thank you very much again, Becky, for kicking off the. Please join me in thanking Dr. Schweighardt. Thank you.